Today's MitoTalk, we are looking at complex 3, Middleman with a double life. Hello and welcome to another episode of MitoTalk Deep Dive, your podcast for decoding the powerhouses of our cell one complex at a time. I'm your host, Dr. Sukant Khurana. Today we continue our journey through the electron transport chain with a look at complex 3, also known as cytochrome BC1. We're calling this episode Cytochrome 3, the middleman with a double life. And for a good reason. This complex doesn't just shuttle electrons, it also flirts with danger, generating reactive oxygen species that can either signal or destroy. Back with us today is the brilliant Basudha Banerjee, who's a budding molecular biologist and expert in mitochondrial therapeutics. Her passion is regenerative medicine. Welcome back, Basudha.
It's great to be back, Dr. Khurana. Complex 3, one of my favorites. It's like the mitochondria's drama queen. Efficient, powerful, but a bit unstable.
Okay, let's begin with basics. What is complex 3? What's complex three's job in electron transport chain?
That's a great place to start. So, complex 3, or rather cytochrome BC1, it generally receives electrons from the ubiquinol, which is reduced at complex 1 or 2, and passes them further to cytochrome C, which then carries them to complex 4. So the transfer happens through a beautifully orchestrated mechanism called the Q cycle, which also pumps protons into the intermembrane space. And this helps to build the proton gradient for the ATP synthesis. So it's both a middleman and an active proton pump.
You mentioned the Q cycle. Can you explain how that works?
Certainly. So the Q cycle involves two electron transfers from ubiquinol. One electron goes to cytochrome C1 and then to cytochrome C, and the other goes via the iron sulfur protein to the cytochrome B, forming a semiquinone intermediate. And this dance results in the translocation of 4 protons per QH2 oxidized, 2 pumped and 2 released from QH2. The net result of this entire process is that the efficient energy transfer, but also a high risk of electron leakage, especially if anything slows down in the chain. And that is where double life begins.
And let's talk about loss generation and its double-edged role. Why is complex 3 a hotbed for loss generation?
Now, that is because of the semiquinone intermediate that is being formed in the Q cycle. If electrons stall or back up due to a block in any downstream complex or say hypoxia, electrons can escape and reduce the oxygen prematurely, forming superoxides. This makes complex 3 a major ROS generator in certain conditions, especially in the heart, brain, or the muscle tissues. It is very fatal. Small bursts of such ROS are necessary for cell signaling, especially the immune and the stress response. But chronic ROS leaks, that's a recipe for oxidative damage, inflammation, and plethora of diseases.
Well, I'd like to pivot to sort of clinical relevance from myopathies to neurodegeneration. What kind of diseases are linked to complex 3 dysfunction?
There are several diseases. Say inherited diseases, like inherited defects in the complex 3 subunits or the cytochrome B can cause mitochondrial myopathy or exercise intolerance and even encephalopathies. Complex 3 dysfunction is also implicated in Parkinson's, ALS, and chronic fatigue syndrome. And remember, drugs like antimycin that can block complex 3. So it's a known target in fungicides and cancer studies.
Well, what's happening in terms of complex 3 therapeutically? What are the drugs and strategies?
With that aspect, currently three major approaches are emerging. So the first one being antioxidants, wherein antioxidants like MitoQ or SKQ1 that localize in the mitochondria to reduce the ROS from complex 3. The second is bypass strategies through molecules like alternative oxidase and from other species to maybe reroute the electron flow. And finally, we have modulators of the Q cycle that try to reduce the leakiness without shutting down the electron flow. See, all this is very tricky. You don't want to block the complex 3 outright. That's a cellular suicide. But you want to optimize its function and minimize the oxidative collateral.
Well, I understand that, and I'm just wondering how AI can help us with all this tricky business. Maybe AI tools are helping with mutation prediction. Can you tell us how AI is helping to study complex 3?
Yeah, definitely. So currently, the AI models they're generally being used to either predict pathogenic mutations in the cytochrome B, or maybe simulate electron flow and ROS escape routes. Alternatively, they are also being used to design the precision drugs that can stabilize the entire Q cycle, or rather the Q cycle intermediates. So right now we are seeing protein structure prediction tools like AlphaFold, which is extensively being used to map how subtle changes in the complex 3 shape leads to diseases even before the symptoms show up. And AI is also helping us design tissue specific interventions by modeling how complex 3 behaves in neurons versus cardiac cells.
So, what's your take home message about complex 3?
Well, that would be that complex 3 is a master negotiator. It balances between energy generation, redox signaling, and mitochondrial safety. It also teaches us that intermediates matter and that any small leak can turn into a big fire if the system is not well tuned. So, mastering complex 3 could give us leverage over some of the toughest disorders, from mitochondrial syndrome to neurodegeneration to aging itself.
Well, brilliantly put. Thank you again, Basudha, to our listeners. If you have enjoyed this episode, do subscribe and share it with your science loving friends. Next time on MitoTalk, we'll explore complex 4, the final flame where oxygen meets its destiny and ATP is born. Until then, I'm Dr. Sukant Khurana. With me is Basudha Banerjee. Stay curious and take care of your mitochondria.
Thank you very much.
Thank you.